Okay, so today we will introduce or we will learn a little bit more about the agents that we have in our network. So we have said systems biology is about agents that interact with each other. So we have different agents, and some of them, for instance, this one promotes this one, but it blocks this one, and it promotes this one, and this one also blocks this one. So there are agents that interact. So which are these agents? So these agents will be they. Okay, so today, for us, these agents will be proteins and genes. Okay, will be proteins and genes. And there can be many more things, but today we will concentrate on these two. So let's go with that. So make it bigger. So there are multiple levels. Here we have the multiple levels. So we have genes. So this is the kind of representation I've been doing. So there are genes that somehow interact with each other, and there are genes, but these genes are translated into transcripts, and these transcripts are translated into proteins, and proteins interact with the substrates and produce metabolites. And at the end, these metabolites. Okay, so these metabolites, all these occur inside the cell. And then the different species, for instance, if you think of the microbiome in your gut, the different species interact with each other. And at the end, all together, they give some clinical trait. So we will concentrate today in these three first levels. Okay, so the central dogma of molecular biology is that the information is kept at the DNA, and then the DNA is replicated by the DNA polymerase. So whenever you need to make a copy of the DNA, you use the DNA polymerase. Otherwise, if you need to translate into RNA, you use the RNA polymerase, and the RNA, messenger RNA, is translated into protein by a ribosome. And yeah, so this is the central dogma, and the information flow would be more like this. But it happens that in reality, that could be some kind of branches. So for instance, some viruses, they go from RNA to DNA, they're capable of doing that. And in the laboratory, you can go directly has been done directly from DNA to protein, or yeah, there are many other possible flows in this information pattern. But the most typical one is this: DNA replicates, DNA is translated into RNA, and RNA into protein. So that is at the high level. So what we will see now is that the devil is in the details. So there are many small details that you have to pay attention. So in general, the way we should think of this is we have the DNA. The DNA, you have double strand, and they contain exactly the same information in a complementary way. So once you are transcribing to produce RNA, what it does is you open the DNA double strand, and then one of the copies is set the coding strand, the other one is the template strand. So the template, the RNA polymerase goes over the template strand and makes a transcript. Okay? And this transcript that is made of nucleotides, later it will be taken by the ribosome, and it will produce a protein. So far so good, but then the devil is in the details. So once you open, there are many possibilities. So one thing is that there are introns and exons. So not everything in the template is translated. So these gaps here, these gaps are parts of the template that are not transcribed. And you can transcribe in one direction or the other. And there are proteins that, or there are genes that use both of them. So a part of the protein, a part of the template can be used to produce a protein, or the same part, but in the opposite direction, can be part of another protein, different protein. And also in one of these centrons, you can have a nested transcription. So you can produce for one of the proteins, this is an intron, so it is not transcribed, but this intron is transcribed for another protein that is read in the opposite direction or in the same direction. So there can be many, many different possibilities. So that is one of the topics. The other thing is that there is not really a single gene. So in the sense that, for instance, all these are, this is a virus, and there are many different, this is a protein of a virus, and the protein, the gene coding for a protein of the virus. And there are many small changes. There are many variants. Okay, so for instance, all these are kind of strains of that virus. And then here you have kind of the canonical sequence, and all these other variants, they have the same nucleotide at the same location, but some of the variants, they may have a change in one of the variants. So the protein produced by this variant will be slightly different from this one. And this slightly different can be, there are silent mutations. So silent mutation is that it is encoding the same amino acid, or it can make a mutation and really encode for a different amino acid. Then this happens within a species, but also if you compare, for instance, this is the phylogeny of coronaviruses. So you see that many coronaviruses here, it is the SARS-CoV-2. So this genome is very much related to many other genomes. So the bat coronavirus is very similar to the human coronavirus, and the human coronavirus SARS-CoV-1 started from a different branch from the SARS-CoV-2. You see, it belongs according to this tree. Something else is how you construct the tree. But according to this tree, SARS-CoV-1 is different from SARS-CoV-2. It started earlier, they diverged before. And for this, one of the tools that we use to construct those trees is this multiple sequence alignment. So you take all the sequences that you are interested in, and then you try to align them and come out with something like this. So which are the differences between the genomes? You may look at the whole genome, you may look at a particular protein, you may look at whatever. Yeah. So more. So we are saying, we are talking about genes and DNA, but in our cells, we have two sets of DNA. One is our own nuclear DNA, that is about 3,000 million base pairs. So 3,000 million nucleotides. It is linear, it is organized in chromosomes, there are instones, they are structured around histones. You have a part coming from your mother, the other part coming from your father. And there is only one copy per cell, but you also have mitochondrial genome. So the mitochondrial genome is not in the nucleus, it is in the mitochondria. And in this mitochondria, there are many different copies of this DNA. It is very small. It is circular rather than linear. It doesn't have histones. It is coming only from your mother. This one has a lot of non-coding sequence. Here, it has very little non-coding sequence. So it is really different from the nuclear DNA. And both, you need both actually. It's not that one is more important than the other. This we have already seen a bit. We have already mentioned in this translation from DNA to protein, there are introns and exons. So exons are the parts that will really finish into a protein. And then when you transcribe from DNA to RNA, you copy everything. But then at some moment, depending on the tissue where this translation is taking place, it knows somehow which are the parts that are needed. So let's say that you are in some kind of cell, and you need this gene, the product of this gene, but the product of that gene in this particular cell is made of these 5 pieces. So you take the 5 exons. But for instance, in some other kind of cell, the exon 3 is not needed. And then the change is that you have 1, 2. So 1, 2 is all this part. And then 3 is missing. So all this part is missing. And then you have 4 and 5. So your protein here in this cell is a slightly, it is sufficiently different from this one. And in some other cell, then you are missing piece number 4. So all these are alternative splicing. It is what differentiates cells. Then also DNA is mobile. So DNA can move across the chromosome, and there are mechanisms to cut a piece of the DNA and paste it somewhere else. And this mechanism is mediated by something called transposomes, and there are many different kinds of transposomes, and going back from RNA to DNA. So there are 4 possible kinds of parts. But the important idea here is that the DNA is not fixed. It's not something that is absolutely fixed, and then it never changes. It can take a while. Also, there are many marks on top of the DNA that also control the translation. So all these epigenetics marks. Have you heard of epigenetics before? No? Okay, so these are chemical marks that you can do on the DNA. Very few of them. So you have DNA methylation. So DNA methylation, you can put, here are the this group. So you put this methyl group somehow associated to the cysteines, to the cytosines. And then you have also acetyl group that you can methylate the DNA, acetylate the stones, methylate the stones. You can also, so by marking this, you can somehow, where am I? Yeah. So by making these marks, you can control which genes are transcribed or not. And for instance, this is very useful during the development of the embryo. So you need, when you are an embryo, you need some genes, but when you are an adult, you don't need them anymore. So you have to mark them to say, okay, these you don't have to transcribe them more in your life. And these marks, they are not for life always. They are marks that are transient, and you can, like with these marks, you can modulate which genes are translated or not. And also, you can, the DNA translation is, or transcription is modulated by transcription factors. So you can somehow interact with those transcription factors. And you do that with this long non-coded RNA. So these are pieces of RNA that interact with the transcription factors. And then they also regulate which pieces are transcribed or not. And also, you have the messenger RNA. And then there are pieces that are called microRNAs. And they control whether this mRNA, messenger RNA, will be destroyed or not. So with the microRNA, you also control what is the protein level that you have at the end. So the concentration of a protein, you can, when we say an agent, we were stating before here, an agent inhibits, so this protein inhibits this one, can be done at many different levels. So it can be done by somehow using one of these marks, or it can be done with, I don't know, some microRNA or some long coding, long coding RNA. So there are many different places where you can do that. Yeah, microRNAs, they are really, really important in many processes. So they control quite a lot which messenger RNAs will be destroyed. And if it is destroyed, it will not be translated. So it disappears. So with the microRNAs, you can control the concentration at any moment. And the microRNA itself is also regulated. So there is a whole regulation pathway there that is rather complex. Then, okay, so in terms of systems biology, it is very important to assign labels to the agents. So you have an agent, so let's say a gene, and you can say where that gene is expressed. So you should expect this gene to be expressed in this part of the cell. So you have all these cellular anatomical entities, and it has here it has about 1 million cellular components. So you can say this gene, this label, this label, this label, this label. And the same for the biological processes that they regulate. They also have about 1 million labels there, and molecular functions. Okay, then there are other ontologies. So an ontology is a dictionary. So it is a dictionary of a controlled vocabulary. So you can you are limited to these labels, and then you can label the genes with whatever words you want from here, but restricted to here. Okay, that is the concept of ontology. And in bioinformatics, it is very important. They are very much used to have structured databases, and there are many different ontologies. So here I'm putting a few of them. And here you can see how they are used. So for instance, if you go to UniProt and you look for a particular protein, then at some point they tell you which are the labels that this protein has. So this protein has a molecular function, it has this one, transferase activity, and it has this one, molecular transducer activity, and so on. So this is for the molecular function, and then for the biological processes, it has also few. It is a way of labeling your entities in databases in a very controlled manner. Yeah.
Depending on the cell, the same link can have different functions.
Yeah. So you put all of them.
You have to put, like, for example, for this kind of cells, details.
Yeah. So in this particular case of gene ontology and UniProt, gene ontology does not differentiate by cell types. So if it has different functions in different cells, you put all of them and you don't differentiate. You cannot say these molecular function in this kind of cell types. As far as I know, there is no ontology for cell types. As far as I know, maybe wrong. Okay, so RNA, there are many different kinds of RNAs too. So there are messenger RNA that we already know, and there is a whole family of so-called non-coding RNA. And the non-coding RNA, you are probably familiar with this one, it's a transfer RNA. So these are the ones that somehow are used to translate the messenger RNA into proteins. And it is done by a ribosome, and a ribosome is mostly made of RNA too. That is the ribosomal RNA. And then there is a whole non-coding RNA that is for regulation. And for regulation, you have long non-coding RNA that is made of more than 200 nucleotides, or a small non-coding RNA. And within the small non-coding, you have also a whole family of things. So you have microRNA, we have already mentioned, but then you also have more kinds of RNA that can be expressed in the nucleus, and then they have different functions. And RNA is a very flexible kind of molecule, can have many different uses. So, for instance, it can be used to make ribosomes, but they can also regulate the expression of genes. So there are something called riboswitches. So if there is this metabolite in the cell, then somehow this RNA takes this shape, and then it interacts with the transcript factor, and something is expressed or something is not expressed if the metabolite is not there. Can also be used to as kind of enzymes, or to translate the packaging. The alternative splicing is also controlled by RNA. Self-splicing, the regulation, and the epigenetic regulation I told you. So they are really flexible molecules. They can do many things. So you can see that the central dogma can add a lot of small details on top of the central dogma. Then RNA structure. So RNA is very flexible in their functions, but also in their structures. So at the end, you will have something that looks in 3 dimensions, like some specific shape. These shapes, sometimes they are very flexible. They can fold in many different ways, and just depending on what is the environment, which are the things that it is interacting with. But typically they form this kind of loops. Okay, so because it is made of so you have 2 strands, and these 2 strands interact with each other, or a single strand that interacts with itself. So it can have many different possibilities. And these are kind of Lego blocks that are used typically in the structure of RNA. RNA is much less stable in shape than proteins. Proteins are much more stable in general. Because of the flexibility of RNA, there are not so many structures of RNA, because it is difficult to see what is the structure when something is changing all the time. And we have said genes are affected by transcription factors. So here is the part that you want to transcribe, and the RNA polymerase somehow will detect the starting codon and then translate. But this finding somehow the starting codon is regulated by a promoter, and then this promoter, maybe it needs many other proteins or transcription factors associated to it, so that the RNA polymerase can see it. So the control of all these complex is also regulated by the cell. It is a way in which you can control which is the level of the concentration of the product of this gene that you have in the cell. And you also have something called operons. So operons are groups of genes that are regulated together. So you have here 3 genes, like Z, like Y, like A, that somehow this is related to the metabolism of glucose and lactose. And then you have some transcription factors, and these transcription factors, they are regulated differently depending on what is the environment, what is the concentration of glucose and lactose that you have. So for instance, if you have glucose, you don't have glucose, but you have lactose, then there's a protein that will join, it will bind this part of the promoter, and then the RNA polymerase will be repressed. And then for these ones, they're not repressed, they are expressed. But then, if for instance there is glucose, then there is another protein that binds here, and then the RNA polymerase cannot start the transcription. And there is no glucose, but there is no lactose either. So you will have this protein here, but also this other protein here. So somehow it has, with this, you see a kind of binary code there. So this is the kind of condition. So if you have something, so for instance, if you have no glucose, this protein will be bound. So this is like 1, 1. That is glucose, this will be 0. And there is lactose. So if there is lactose, this is 0, but this is 1. So difference, okay, if this is, if there is no lactose, this is 1. So somehow you have here a binary encoding of what is your environment. And this binary encoding is achieved by the transcription factor. So by regulating the expression of all these genes at the same time. So openers are groups of genes. And what else here? Yeah. Also, more about regulation. So you can also regulate by phosphorylation. So let's say that you have some, where is that? So that the transcription factors that you need, you can phosphorylate or not. And then by this phosphorylation, you are somehow encoding also what is the environment. So whether you have lactose or whatever you are looking at. And then the transcription factor will not help or will regulate. The regulation of a gene expression is not always yes or no. It can also be at different levels. So can be more or less efficient depending on the phosphorylation state. And there is a whole hierarchy of transcription factors. So this idea of operons, there are 3 genes or a group of genes that can be transcribed together. You can do it at multiple levels. So you can do it at a very low level, and then this transcription factor is regulating a few genes. But then this transcription factor is itself a protein. So it has its own transcription factors. So you can put on top of that, you can put another layer of regulation. And on top of this, you can put another layer of regulation. So you can take this idea and create multiple levels of regulation. So it is the regulation of the transcription factor. And here you have a paper explaining how they measure all these regulation things. Then there are kind of regulation motifs. So regulation motifs are typical patterns that exist in these networks. So you have a typical pattern is this one. So you have a protein, or here it is represented the messenger RNA, that self-promotes itself. It is also promoting another protein, but then this protein is inhibiting the first one. And there is another pattern that is with 3 genes, that is this one. So this promotes this one, but this inhibits the first one. So this kind of regulation patterns, they are called negative feedback. So it is if when something goes high, so if this goes high, this goes high, this goes high, but then because this goes high, it tends to make this down. Okay, so you are trying to regulate at a particular level. So this kind of homeostasis thing or every time you want something constant, somehow you have some kind of this pattern. And how to measure gene expression. So there are many different technologies to measure that. Here I'm just mentioning them. And particularly important now, I would say this one, RNA sequencing. Flow cytometry is also very much used. So these are mostly, RNA sequencing is for many genes at the same time. Flow cytometry is when you want to look at many cells at the same time. And all the others, they are more specific for a particular gene. This is more or less kind of summary of these ones. For instance, for your practice that you have to do, you may want to talk about these technologies if you want. Okay, so genes are translated into proteins, but not everywhere. So it depends on and not all everywhere and not always. So the where and when they are also very much regulated. Here, what I'm showing is the development of an insect. So here, this virtual embryo, it is of Drosophila. Drosophila is this fly, small fly. And these are different proteins. So where this protein is expressed at a particular time is expressed here, but it is not expressed there. And you may here see some lines. You know that insects are made of segments. So these are the segments. These are the preliminary parts that will later give rise to the segments. So there are specific patterns, and so spatially, so in terms of space, they are very much regulated also. And they are regulated in space, but they are also regulated in time. So if you look at sometime later, and you look, for instance, for this protein has a different expression pattern some other time. So they are regulated in both in space and time. Then something that is often used with gene expression is to measure, to compare to different conditions. So let's say that you have some healthy people and you look at what is the gene expression of a particular gene. So these healthy people, they have a gene expression, they don't all have this exactly the same amount, but they have somehow kind of distribution. And when you look at disease people, then what you see is that this gene has changed. Many other genes, they have not changed, but some of them, they have. So the idea of this differential gene expression is, okay, we have the 20,000 genes, which are the genes that are differentially expressed between different conditions. And then here you have the different, you have all the genes, different kind of cells or different kind of conditions or whatever, and then you can try to separate them. So in these conditions, so let's say this is healthy, this is diseased. So in the healthy condition, all these genes are upregulated, so they are particularly expressed. These ones are downregulated, so they are particularly inhibited. But in the disease, it's more random, but it tends to be the opposite. So this is more inhibited than it should, and this is more activated than it should. So finding these genes that are differentially expressed in the different conditions are yeah, and expression is normally measured in fold change. So fold change is a measure of the following. So you have two conditions. You have condition one, condition two. And in condition one, you have a distribution like this. So let's say this is an expression, let's say expression one. And in condition two, it is displaced. So we have condition two. and then you compute the ratio. So the fold change. The fold change is E2 divided by V1. And these are distributions, so you may divide the means, you may divide whatever you want. But this is the ratio between these two. So this ratio, let's say that there is no change in this. There is no change in this gene for these two conditions. So this will be 1. And then if you take the logarithm, because these things are multiplicative, it is better to take the logarithm. The logarithm of this is 0. So that is why you have here that the logarithm in base 2 of the fold change is centered around 0. So here you have the fold change. So these ones are inhibited, these ones are promoted and overexpressed. And here you have the p-value. So the p-value is, as I told you before, these things are distributions. So how sure you are that you are really away from 0. And you can also see the p-value for that. So what you want is to be very sure. So this is minus the logarithm in base 10 of the p-value. So the farther you are, the more sure you are in what you're saying. And the farther you are away from 0, the more different those two conditions are. So this is very much used in many research labs. I mean, yeah. But when you are studying any disease, this is very much used. And there will be genes that do not change at all. So these are the housekeeping genes. So the housekeeping genes are genes that if they change their expression, the cell dies. So they are not viable anymore. So there are many genes, I don't know, constructing the cytoskeleton. If you cannot construct the cytoskeleton, you have no cell. And these housekeeping genes are genes that do not change. So yeah, they are stable in expression. They are somehow related to the cellular maintenance. They are essential. They are conserved among different species. And these set of genes, there are a few thousands of them, they don't change from cell to cell. And those genes are typically used to normalize the data because when you read, you analyze this, what is the gene expression by any one of these means. It depends a lot on the specific experiment that you do. So you do some experiment today and you get some readouts, and you do it tomorrow and you get some other readouts. And so the specific numbers are different, but you know that for the housekeeping genes, they cannot change. So what you can use is what is the expression level of the housekeeping genes, and then I will use that expression level to normalize the data to make the data comparable between different experiments. For instance, yeah, okay, so now you have some genes that are different between two different conditions. So what do they mean? So now you use the ontologies to try to understand what they are doing. So let's say that you have two conditions. So these two phenotypes, A and B, and you have here in A, all these genes are down-regulated and these are up-regulated, and in B it is the opposite. So now, how can I know which are the functions of these genes that are different? So now, because these genes have these biological process annotations from gene ontology, you can see, okay, which are the labels that are overrepresented in one of the groups and underrepresented in the other group. That are different. So there is a statistical significance between the number of times I would expect that label to happen if that happened randomly. So which are the ones that are not random? And we may come out with some set of labels that are different between the two conditions. And the same for pathways. So you can do the same idea with pathways, and not with labels. But which are the pathways that are overregulated or repressed? So overexpressed or repressed. Yeah, so here there is a paper in which they somehow combine all these ideas, and they bring some others like, okay, you can look at gene ontology, you can look at care, or you can look at the reactome. Care and reactome are two different database pathway databases. You can see immunology, or you can see whatever you want to see which are the differences between these two sets of genes. And you may look for what they are called biomarkers. Biomarkers are these genes that are different here, different between the two conditions. So I have healthy people, I have person with disease, and then I see that this gene is overexpressed here. So this overexpressed in the gene B is overexpressed in one of the conditions. And then I know that this is the difference between the two. So what do you use that difference for? So you can use it to classify patients. So there are many diseases that somehow the genetic signature is different between different variants of the disease. So you can see specifically you have this disease of type A, of type 2, of type B, and so on. Or you can use it to predict what will happen with that patient. So you classify the patients according to how they will respond to treatments, or how they will evolve with time. So there can be different uses of these biomarkers. And biomarkers, we are presenting them as associated to genes, but they can be associated to anything. So they can be associated to, for instance, here with these, they are epigenetic non-coding RNAs, but you can also have somehow epigenetic biomarkers or metabolic biomarkers. Okay, what else? There are many databases of this kind of stuff. So array express and gene expression omnibus are the two most known. These also are very well known. European Nucleotide Archive. So those are databases that contain gene expression data. Then software to analyze this kind of stuff. Software. You can bring this this analysis one level up. So, for instance, in our gut, we have many different kinds of bacteria. So all these bacteria, they all have their own genes. So you can do the analysis, but not now for a single individual, that will be a single bacterium, but for a whole set of bacteria, and then this is called metagenomics. So you analyze all together. What is the difference between the microbiome of a person and the microbiome of a diseased person, healthy and diseased? So these metagenomics approaches, they are now very in fashion too. You can also do phylogenomics. So you can analyze the whole genome of different related species and see how they are related. So when one evolves from the other, how separate they have been in time. Any question up to here? No? So I think it is a good time to make a break. So we make a break of 10 minutes. Venga. Any questions regarding the gene? No? So if there are no questions, then we move to the other kind of important actors that are proteins. So proteins, normally we tend to think of the protein isolated in the cell doing its stuff, But reality is more like this one. Actually, this guy is very famous for making this guy, David Goodsell, is very famous for making this kind of representations, and they're quite kind of beautiful. So the cell is full of stuff. There is very little space, and they have to perform their functions in this environment. Okay, so what the protein is? Well, this you already know. So you have amino acids, and amino acid looks like this. So it has a backbone that is all the time fixed, and then there is a variable side chain. And then this is a single amino acid. When you put two amino acids together, you have this peptide bond here, and water is released. And then you have now a backbone here. So this backbone, this amino acid, this carbon here is called the carbon alpha. So you have a carbon alpha, and if you have more, then you would have the ones that are coming from that are just beside the nitrogen is the carbon alphas. And sometimes proteins are represented only with the carbon alpha. So you forget about everything else and you only put the carbon alphas. So depending on what you want to do with the protein, sometimes it's represented like that. And then you have the residues, and here the residues are represented there. And there are many different kinds of residues. So this is the fixed part. And then depending on what is the residue, so these ones are electrically charged and they are positive. So they have a positive charge here in the nitrogens there. And then these other are negatively charged. Some others, they are polar, so they like to be in water, but they are uncharged. And these ones, they are special with respect to all these properties. Some others, they don't like to be in water. So all these hydrophobic side chains, they are normally they like to go into the lipid membranes. And you can analyze many different properties of amino acids. So depending on what is the residue, you have different properties. So they are aromatic, so they have cycles or chain that makes a cycle, aliphatic, that is amine, hydroxylic, gyne, small, acidic. So you can have many different properties. Normally, amino acids, they are represented by a single letter. There is a kind of standard code. And then proteins adopt some structure in 3 dimensions. But this structure in 3 dimensions, it is this one, tertiary structure, but there are 2 levels below and 1 level above. So the 2 levels below are, you start with a sequence of amino acids. This sequence of amino acids, they may locally form this kind of shapes. So this is an alpha helix. These are beta terms, so beta sheets. So you have a kind of sheet that is called the beta sheet, and the alpha helices. So alpha helices and beta sheets are the secondary structure. So here you have the beta sheet, you have alpha helices there, and all together with the terms, they make the tertiary structure. But then once you have the 3-dimensional shape of your protein, it may bind some other form a complex. And this is the quaternary structure. There are a number of forces that make this structure stable. One of them is obviously the peptide bonds. So this is these ones here, they somehow contribute to the final structure, but also van der Waals interaction. So this region interacts with this one, or hydrogen bonds that this residue here interacts with this one, or ionic interaction. So this residue here interacts with this has a negative charge, this has a positive charge. They interact with each other, or sulfide bonds or covalent bonds or whatever. And there are patterns, so there are kind of motifs that are regularly seen in proteins. So for instance, this is one of them, beta alpha beta or beta barrel or whatever. So there are patterns that are typically seen very often. These are the different portions again. So the structure is stabilized by the peptide bonds plus all these things. So salt bridges or hydrogen bonds or hydrophobic interactions. So for instance, if you have a set of amino acids that don't like to be exposed to water, they may close, they may try to form an hydrophobic core that is not exposed. And then these van der Waals dipoles or disulfide bonds, or you have a metal ion and then it is somehow interacting with all these amino acids. And once you have the structure, so the structure may look like this, it has different properties. And you may look at here, I'm just representing two of them. So one is hydrophobicity, whether these particular regions, they like water or not. So these regions, they may not like water, and these other regions, they may like. Or what is the charge? So some parts are neutral, some parts are negatively charged or positively charged, or whatever. Also, the charge and the shape, because the charge is one important part that defines the shape, may depend on the pH. So if you put a protein at neutral pH, it has some charge distribution, but if you change the pH, some of the residues that were not charged, now they become charged. And then the charge, the surface charge changes, and then the protein changes. So the shape changes. It may also depend on salt concentration. So you have this protein here, and you change the concentration of salt in the environment and in the solution, and then, for instance, here for this particular protein, if you have very few salt, then it folds like this. If you have a lot of salt, it doesn't fold. So it depends on the protein, what is the behavior. But the important thing here, the pH and the salt concentration may change the shape of the protein. Also, some proteins, they like to, they fold very easily, like this one is folded, but some others, they, depending on the environment, they may become quite disordered, and then you can have a whole range in between. So parts of the protein are folded, but some other parts are not. There are, so this may be kind of pathological behavior, so it shouldn't behave like that, in the sense that the useful protein is this one, and all these other ones are artifacts that should be destroyed or refolded or whatever. But there are proteins that work like this. So some proteins these are called intrinsically disordered proteins. So some proteins stay normally like this, and then when it binds to something, it folds. But only upon interaction, they work like that. So they may be an artifact, or they may be the normal behavior.
They're not bound to something. There are something or no.
That
Active.
depends. It also depends on the protein. So there are proteins that are when they are bound, they are inactive, and then when it is part of the regulation. I may inhibit it and intrinsically dissolve the protein by binding to it and making it fold. And all these folding and folding is controlled by an energy landscape. So depending on what is the particular shape, you may compute some associated energy. So for instance, here you can think of an electrostatic field. So I have a part of the protein that has negative charge, the other part has a positive charge. So depending on what is their relative orientation, they will have a different potential. And then this potential is an energy landscape. So maybe this energy landscape, it has this one particularly, it has two stable or three stable structures. So one fold, another fold, the same protein in two different shapes, and another one that is less stable. And depending on where you are, you have, for instance, a region that is moving to this side or to that side or whatever. And also, because of thermal energy and also because of interaction with other proteins, you may go up in this landscape. So it is not that once you hit the minimum, you stay there. So you have energy in the surrounding, and then in this energy, thermal energy, collisions and so on, can make you go up in this landscape. So you are all the time moving. And that is natural for proteins. They have to do that. Then proteins are flexible objects, so they can adopt many different shapes. And we know now that they are related to this energy landscape. And the energy landscape and the shape may depend on what it is interacting with. And there are kind of two different kind of interactions. One that says, okay, you have some shape, and this protein has to interact with this substrate, and then this substrate has the right shape to interact with it. Or it might be that at the beginning the substrate doesn't have the right shape, so this receiving pocket doesn't have the right shape to interact with this. But when the substrate approaches, then the shape here changes. This is called the induced width hypothesis. This is called the lock and key hypothesis. So this is like a lock, this is the key, and then they interact. Or you may change your shape as the ligand of the substrate approaches. Very likely this is a more effective representation of reality. So things are not fixed in general in life. And also you have you interact through sites. So these sites can be the binding site, the catalytic site. There are many allosteric sites. So there are many different sites. These sites typically they are kind of pockets that are somehow convex, so that you can hide a bit the interaction. So it is not typically they don't interact so much on the surface in this kind of more protected areas. And you have here allosteric sites. So let's say that your protein is an enzyme. This enzyme has to interact with the substrate here. This is the active site. But then there is another region that regulates this interaction here. So if something is bound, it has the right shape for interaction. But if something is if something is unbound, it has the right shape. But if something is bound, then the shape changes. The shape changes, and then the protein is inactive. And you can imagine that could be several allosteric sites. This is an inhibitor. You can have positive modulation rather than negative. So when it is bound, it is when you can thrive. You can have many different possibilities. So this goes into the direction. These agents that are interacting between them, you can regulate them a lot. So we have seen all the gene regulation. Now we have the protein regulation. And the folding, we have already mentioned this idea. It depends on an energy landscape. So when it is properly folded, you are kind of in a local minimum of this energy landscape, which is a good minimum in the sense that if you go out, you are forced to go typically the direction of the minimum energy, so you will reach fold again. But you can have folding intermediate, so you can have something that is not properly folded, but somehow you will fall because you are allowed to go up because of this thermal energy. So because of this thermal energy, you are allowed to go a little bit up in this landscape, then you will fall to the right ones. Some other times it is very difficult, and then you need the help of some other proteins to help you fall. These are called the chaperones. The chaperones help you to find the right minimum energy minimum. Sometimes you have aberrations. You may have aggregates that are in the local minimum that somehow you will not find the right shape ever. And then you have to destroy these and then re-express the protein and see if it can fold properly. You can have oligomers, so they bind to similar proteins or to other proteins, and then they find their minimum, their shape. You may have these amyloid fibrils, so they're kind of tubes where you have repetitions of the protein. And actually, Alzheimer is somehow related to amyloid fibrils, fibers. Then proteins are not only amino acids, but they also have post-translational modifications. So once you have the protein, now you can modify it in multiple ways. So you can, for instance, put a methyl group, or you can oxidize it, or put a nitrogen, or phosphorylate it. And here what you have are the different enzymes that do that, and which are the amino acids that are. So for instance, if you want to phosphorylate, you can only phosphorylate these three amino acids. And yeah, there are multiple modifications. So again, the idea of regulation. So the protein is not just the protein, it is the protein modulated by many other things. And then proteins have to perform their function somewhere. How do they know where they have to go? Here you have the synthesis of the protein is here at the ribosomes, but then it goes through the endoplasmatic reticulum, and they are put into vesicles, and these vesicles, they are sent to the place in the cell where they have to go. So somehow, when you translate the protein, you have to make some marks, and these post-translational modifications are part of the marks that are used. Or you may add a small piece at the end, and this small piece at the end is somehow a message to the vesicle to say where you should go, and then you can set it to anywhere in the cell where this protein should go. This process is called sorting or targeting. Also, there is the concept of recycling. So we have seen it a bit here. So let's say that we have translated the protein, but for some reason it cannot find its right conformation. So what do we do with that? It is so for the cell expressing something is cost a lot of energy, and if I cannot use this thing, that is useless. So I should recycle that. And there is a whole process of recycling, recycling misfolded proteins, recycling useful proteins, but that they might be damaged for some reason. And then there is a whole, so once you translate, there is a whole pathway taking care of which proteins should be destroyed and reused to take the amino acids again and form new proteins. Or let's say, for instance, that you are in some condition. In this condition, you need this kind of proteins, so you express them. You have now these proteins, but now this condition disappears. What do I do with those proteins? They are correct proteins, but I don't need them anymore, so I have to recycle them again. and there is a whole pathway taking care of that. One of the two important agents here are this proteasome. The proteasome is one of the main proteins that are or complexes that are recycling, and this phagosome. Okay, then the structure. The protein structure is very important. So protein structure. Here I don't know why I have this slide. We have already seen protein structure. So, okay, proteins alone are not proteins are not the only biochemical entities that we have related to amino acids. So we have many other things. So we have, for instance, lipids, and there is a whole branch. This is called lipidomics. So lipidomics is what kind of lipids you have, and there are many different kind of lipids. And here you have lipid structures that somehow store lipids, so chylomicrons, so low density. This is cholesterol, low density cholesterol, high density lipids, and low density lipids. So you have to store the lipids somehow, and these are some molecules that are related to this lipid and to this lipid storage. And then there are proteins associated to this lipid storage. So part of this structure is made of proteins. You also have proteins somehow linked to sugars, these peptide glycans. So it is a part. It is a part of protein, this one, but then you have a glycan on top, or you have a sugar on top. And these are the ones, these peptidoglycans are the ones that make the membranes of many bacteria. So here you have, this is the cell membrane, if you want, the green one, but then outside you have a peptidoglycan shell that protects you, or you may have multiple peptidoglycan shells. So you have, for instance, you have the cell membrane, and then these peptidoglycans, and then you have phospholipids, so it is lipids, and then you have sugars outside. So you have all kinds of structures. And here you have, I told you before, the structure depends on the energy, but proteins are a flexible object, and as such, they need to be flexible. So it is not a flexibility and movement, something bad. It's something that we need. So here you have.
Vale, 50% de descuento en Burger King.
So for instance, this is the life cycle of SARS-CoV-2. So this is how the cells look like, and everything is made of proteins, these glycans, these lipids. That is the cell of some virus. And again, the virus is made also more of proteins, but these proteins are protected by glycans. The green points are glycans, and somehow the glycans are meant there to hide from the immune system. The immune system is very good at recognizing amino acids. This nucleocapsid, it has proteins and RNA. So this is a protein, and then this protein has to. So this is a sugar. So there are a lot of interactions with, this is the cell, our cells. And then the spike recognizes which this is the cell. And then once it recognizes, so all these movements, you are moving along this energy landscape. So moving along the energy landscape is something that you need. So these things are, so all the movement, everything that you see here, this is the right, this translate. So all this is moving along the energy landscape. So movement is something that is intrinsic to life. Okay, so you can watch the video by yourself. Right, this is very interesting. Okay, so how do you determine the protein structure? So there are mainly 3 experimental techniques. One of them is X-ray crystallography, that traditionally has been the one that has produced more structures, but it requires the protein to be crystallized. So a crystal is a regular sorting or arrangement of this protein, and not always you can crystallize. Actually, it is not that not always, it is that seldom you can make it crystallize. So you have to try many different conditions until you can crystallize it. And nuclear magnetic resonance, you don't need them to be crystal, but then because of the kind of measurements you are doing, you are very much restricted in the size of the protein that you can address. So typically, you can see proteins whose weight is less than 30 kilodaltons. And then you have cryo-electron microscopy, and this cryo-electron microscopy doesn't require crystallization. You can address very large complexes, and also you can see all the dynamics. So they're frozen, but you have many copies of the same thing, and then you hope that each one wasn't a different conformation. Then you can also predict the structure. So it is not only that I can measure the structure, but you can also, given the sequence, you can try to say what the structure is. Have you heard of AlphaFold? Yeah. So AlphaFold was a kind of revolution a few years ago, 2-3 years ago. And it was because they were using deep learning to predict the structure, and they used how they converted the alignment problem into an image problem. It was really interesting, and using these neural networks and multiple sequence alignment. So you have a structure, you see which other proteins in other species are similar to yours in sequence, and then from this information you are able to predict the structure. This has been possible because there has been many structures solved experimentally. AlphaFold has been a revolution. It is very useful, but it doesn't produce perfect answers. So experimental structures are still better than predictions. And then proteins can do many different functions. So they often act as enzymes, and you can watch the video by yourself. So you can see how this chemical reaction here is somehow facilitated by the protein around. And that is why otherwise, you can imagine in the cell environment that is so crowded, these 2 molecules would seldom find each other in the right orientation. So the protein somehow helps this molecule to be in the right orientation, this molecule to be in the right orientation, and then the chemical reaction occurs. And they are transporters and the carriers in cell membrane. So here, for instance, when you want to export something or to take something from outside the cell, so you can make pores, and transfer whatever you're interested in. And here you see, for instance, this is the cell membrane. The cell membrane is made of lipid. So all these transmembrane regions, they're made of hydrophobic amino acids, while the parts that are exposed, they are made of hydrophilic amino acids. You can watch the videos by yourself. They are also involved in movement. So, for instance, the flagella of bacteria or sperm cells or whatever, they are made of proteins that somehow they work as a machine and they make the flagella move. And muscles and transports along the so you have a vesicle, you have to send it to some part of the cell. So they are really, this one you can see, it's really interesting to me. The other ones are not very interesting, so it is not that they are not that. But this one is amazing. You can see it walking. This is a microtubule. You can see the protein walking. They're in the
Yo sería estos 20 árboles.
sí.
Oh, bueno. Hecho la mano.
Bueno.
Ya, vale.
Ahora. Sí, see, walking. And this is the energy landscape. You're seeing the energy landscape. And to move in the energy landscape, you need energy. So ATP, all these things. This is the cytoskeleton. Okay. What else? Yeah, antibodies are also made of proteins. And proteins, we have seen this thing that we have about 25,000 genes. But when you translate, because of these alternative splicing and post-translational modifications, we can make from 20,000 genes, we can make 1 million proteins, different proteins. So actually, in number of genes, we are not, we don't have actually so many. There are many plants, many fungi that they have many more genes than we do. Okay, so here is a little bit how you measure the levels of proteins that you have. So how much protein do I have of a particular kind, or how do I measure post-translational modifications? So there is a whole range of techniques. These are experimental techniques that allow you to measure those. And if you are interested, you may go to that paper. Yeah, and again, we have the space and time regulation of the protein structure, of the protein expression. So, for instance, you may look at a particular protein. This particular protein, this is this histone H2B to the arrow. Okay, so it is distributed. And these other ones, it is distributed there. And these are different kind of microscopies to look where proteins are, but they are not everywhere. And they are, so the regulation of where they go and when they go there is very much controlled. And yeah, these are interesting. This is another interesting idea. Let's say that I'm interested in seeing where this protein goes. How do I see it? How do I see the protein? So how do I know that is the protein I'm interested in? And the trick is this one. So this is the protein that you are interested in. And then on one of the sides, so here the N-terminus of the protein. So in the N-terminus, you link with something that is fluorescent. So this one is the green fluorescent protein. So when I illuminate with a particular wavelength, what I see is this one. It is not this one, but because I know that the green fluorescent protein is linked by construction. So in the gene, I put the two genes together. So I have modified cells where the gene of this protein is modified to also express the green fluorescent protein. So what I see is this one. It is not this one. And then proteins interact with each other. So you have protein A, protein B. Here they are, of course, subunits, but the concept is the same. And then the kind of interactions are the same ones that we have seen for the protein structure. So some amino acids don't like to be exposed to water. So if you have some hydrophobic residues here, some hydrophobic residues there, they will tend to go together because they don't like to be exposed to water. Or you may have salt bridges, or you may have hydrogen bonds or whatever. So they are the same concept that we have seen for protein structure. And also this idea of induced fit also applies. So maybe these two surfaces have to be complementary to each other. So they have to somehow go well with each other. And the shape itself may also change upon binding. Here are techniques to analyze, experimental techniques to measure protein-protein interactions. There are a lot of databases. Maybe one of the most famous ones is this one, STRING. And for proteins, you may describe, again, as we did with ontologies, what is their function. So their function at the end, well, this is a lot of text, but at the end, you assign a label. And these labels are from a controlled vocabulary. So this one, this is the enzymatic code or something like that. I don't remember what EC means. It is something like that, enzymatic code. So at the end, you put labels that are coming from control vocabulary. And this control label may translate it to the different databases with different names. Typically, if they want to be consistent, they will use the same one. So if this is 2710.1, they will keep the same number in the database, but they may not. And there is a whole range of cross links between the databases. Then you can also annotate the functions with, this is enzyme commission numbers or the ontologies, or there are many other ontologies that you can use to label what is the function of your proteins. And then for many proteins, you don't know what the functions are. So there are many different ways to try to predict what the function is. For instance, a very simple way would be, okay, I know the sequence of this protein. I don't know what this does, but I know what this does. And I know that in sequence, these two are very similar. So probably the functions of this are also the functions of this one. So that is a very simple way. This is I call this sequence similarity or structural similarity. Yeah, there are many different ways to try to predict, but this is the real problem now. And then proteins participate in pathways. And then you have, for instance, this is a particular protein. This one is protein participate in all these pathways. But it is not that they do a single thing, but they participate in many things. And then a pathway looks like this, but that's the networks that we started from. So we say we have agents that interact with each other. There's databases of these pathways. And this could be. Any question?